A very warm welcome to all the dignitaries present over here. I am J. Komal from the Division of Entomology and I express my sincere gratitude to everyone for providing us this opportunity to present our work at such a prestigious platform, and it's an honor for us. Firstly, I would like to give a brief introduction about the group of insects that I have been working on, which is the moths. Moths belong to the order Lepidoptera and their significance is marvelous. They account for 10% of the biodiversity, and even though their efforts in pollination are always underappreciated when compared to the bees, they are very important nocturnal pollinators, imparting their ecological significance. Economically, they have paramount importance as 90% of them are phytophagous in nature. This major group is informally divided into macro and micro Lepidoptera based on its size. The former contains moths having a wingspan greater than 20 mm, while the micro moths have a wingspan less than 20 mm. Why are these tiny micromoths important? It is that the micromoths constitute 40% of the Lepidopteran species and play vital roles as pests and pollinators. Due to their internal feeding habits, they are generally found as stem borers, leaf miners, etc., being closely associated with the host plant, making its study and documentation important. Based on this importance, taxonomic studies on Microlepidoptera of Pusa campus, New Delhi has been conducted. The reason behind choosing Pusa campus is that it has a large area with year-round vegetation harboring several pests. So, in the context of exploration of micromoths, it is definitely an unexplored treasure. If we analyze the research gap, there are limited taxonomic studies on Indian Microlepidoptera, while the micromoths of Delhi being unexplored, and there is no systematic assessment of the moth fauna of this region. Considering these lacunae, two objectives were framed, which is to prepare an annotated checklist for the moths of Delhi and to conduct biosystematic studies on Microlepidoptera of Pusa campus. For the checklist, a huge data is collected by an exhaustive exploration of the museum holdings of the National Pusa collection, where 1998 specimens dating back to 1907 till date are studied. Field surveys were conducted by setting up light traps in the following sites, accounting for 25 survey nights. Also, infested plant parts were collected to rear them in order to document the different life stages of moths for a host-based study. All the moths are processed finely by the established procedures and then dissected for slide preparation. Later, photography was done using an auto-mountage microscope. Apart from all this, the data was also procured from various citizen science portals. Data from all the previous literatures and isolated publications were collected, and finally this huge data is refined by taxonomic updation. The output of such an extensive data compilation is an updated systematic checklist comprising of 331 species of moths belonging to 32 families under 14 diverse superfamilies. The checklist was arranged hierarchically from superfamily to species level and alphabetically, including its common name, synonyms and occurrence records. This adds a huge number of 239 species as new records to the existing moth fauna of Delhi. If we analyze the checklist of the 14 different superfamilies, Noctuoidea was observed to be the most numerous one with 164 species, followed by Pyraloidea, whereas the superfamily Calicoidea was the most diverse one with 8 families. Of all the 32 families, Erebidae stood to be the most numerous one with 93 species. Although the number of micromoths accounted for one-third with only 98 species, the diversity of these tiny moths was way too higher than the macromoths. Illustrations of 195 species were provided, which aids in pictorial identification of the moths of Delhi. The degree of versatility of moths was highly significant, ranging from the most beneficial pollinators to the most destructive pests. Although being pests in immature stages, the hummingbird hawk moths, which include Hylas lineata and Macroglossum species, are the most efficient pollinators of various crops due to their diurnal nature in contrast to the nocturnal character of moths. Some species such as Cephanodus hylas and Acherontia stix are of interest, as the former is a pollen robber and the latter being a honey robber. Apart from the pests of agriculture, horticulture and silviculture importance, a parasitoid Epigania melanoleuca, which effectively controls the sugarcane leafhopper, is documented. Some surprising records of species include Cunugia latipenis, which is known to be found in higher altitudes, was also observed in the present study. For the taxonomic studies of micromoths pertaining to PUSA campus, 37 species under 34 genera were studied in depth, belonging to 8 families, of which 28 species are new records to Delhi. Illustrations were prepared for all the 37 species which are economically important pests, such as the Phycodus radiata, which is a leaf roller of ficus species, while Hypertimus pathota is a pest of mango, and Copamintus being a leaf webber of the tree species Acacia fistula. The other pests include Dudua aprobola, the mango flower webworm, and Lobosciza coinigiana, which is a leaf webber of neem, and a beneficial weed killer such as Bactria venosana is also studied, which is a stem borer of weeds like cyperus species. 15 species of crambids were studied, of which 11 are important pests of rice, and surprisingly, 3 additional species of little known rice leaf folders were also studied. Among the pyralids studied, Hypsopygia mauritialis is an inequilian in the wasp nests and is known to parasitize Vespa species, being beneficial to the honeybees. Variations of different morphological characters were studied, wherein the structure of labial palpi differed even among the members of the same species owing to its sexual dimorphism. Redescriptions of species was done with emphasis on male genitalia, and these are some of the important characters which were used for the description of genitalia. Male genitalia was studied where the variations were observed to be species-specific. These are few more illustrations of the same. The species-specific variations were explicitly notable in the female genitalia too. Biology of some economically important species was studied in the laboratory, and all the immature stages along with its symptoms of damage were documented. This facilitates identification and detection of the pests at an earlier stage. To summarize, the salient research findings of the present study include an annotated checklist of moths of Delhi comprising of 331 species belonging to 32 families under 14 diverse superfamilies, adding 239 species as new records to the existing moth fauna of Delhi. Illustrations of 195 species provided which serve as an aid for the pictorial identification of moths of Delhi. About 2,686 specimens were identified, studied and augmented to NPC. In-depth taxonomy of 37 species belonging to 34 genera under 8 families was studied, of which 28 species are new records. Illustrations of taxonomic characters studied are provided, and biology of some economically important pests are studied, including the documentation of immature stages along with its symptoms of damage. We have registered our database of specimens studied with GBIF to be accessible for increased viewership. I have published 2 research papers, one in Biodiversity Data Journal with a NAS rating of 7.22 and the other one in Current Science with a NAS rating of 6.73. Another research paper has been communicated to the Journal of Asia Pacific Biodiversity. My other publications include a popular article and an abstract of my work. I am so delighted to share that our effort was recognized and broadcasted by the Times of India and the Indian Express following our publication of Moths of Delhi. For presenting our work on Moths of Delhi, I have received the Best Oral Presentation Award, and these are some of the other curricular accomplishments, and I have even qualified ICR NET and ARS prelims too. I wholeheartedly thank my chairperson, co-chairperson and members for supporting me and guiding me throughout the journey. I extend my sincere gratitude to the respected Director and Dean, Professors, Scientists and staff of Division of Entomology and PG School and all my dearest friends and seniors who have always tried to bring the best version of myself. Finally, I would like to end by quoting few words of the great taxonomist Linnaeus that if you do not know the names of the things, the knowledge of them is lost too. Thank you.